So today, we travel together through the wonderful world of the cell. Just like before and just like every other time on Blackboard, these are just reviews. It's not everything. It's all the key stuff in case you need a refresher. So in lecture, we talked about this, the fact that the cell theory tells us that the cell is the structural, functional unit of a living thing, and that there are size limitations to cells. And here's what that means. The bigger a cell becomes, the larger its volume relative to its surface. Well, the bigger the volume becomes, the more metabolic function that cell has to carry out. So from this cell to this cell to this huge cell, this cell, number 3 here, has the greatest metabolic activity because it's got the biggest volume. It's got more organelles in it. Well, the more metabolic function a cell has to carry out, the more it has to rely on transport of nutrient and waste across its cell membrane. Well, even though to our eyeballs that cell there looks like it would have a huge cell membrane, it actually doesn't. The bigger a cell becomes, the less surface it has relative to its volume. So it turns out cells naturally can't get very big because they can't have enough cell membrane to support being that big. Cell theory also tells us that cells can become specialized and that in a multi-celled creature like us, they are highly coordinated and they have the ability to recognize and communicate with each other. At the very basic, cells are always a plasma membrane or cell membrane, cytoplasm with things in it, and then a nucleus. Plasma membrane is for boundary, support, and recognition and transport. The cytoplasm is the intracellular fluid, the cytosol, with everything else dissolved into it. Cytoplasm is affected by osmolarity. Cytoplasm contains organelles and structural proteins and enzymes. Cytoplasm is where a lot of chemical steps in metabolism take place. Then we have the nucleus, which contains the DNA, and you make RNA from that DNA. DNA is the user manual for the cell. So it is the site of control and regulation and response. We focused on the plasma membrane in lecture and its structure and how that structure relates to its function. The plasma membrane is a phospholipid bilayer. You should be able to circle those on this picture. 2 layers of phospholipids. Well, phospholipids are both polar and nonpolar at the same time, or hydrophobic and hydrophilic at the same time. Hydrophobic means water-fearing, and that would be the fats. Hydrophilic means water-loving, and that would be the polar head. In lecture, we talked about how that structure affects membrane permeability. Because of the way it's arranged, and because of the fact that you have cholesterol every now and then instead of a polar phosphate head, molecules that are lipid or lipid-derived, molecules that are nonpolar, are soluble across the cell's membrane. They'll come in on a cholesterol, go through the lipid tails, and come out on a cholesterol. So they're soluble across the membrane on their own. Molecules that are charged, or polar, or hydrophilic, cannot cross the membrane on their own. They'll come in wherever there's a phosphate group, but they get pushed right back out again by these fatty acid tails. So that means if the cell wants to transport across the membrane a molecule that's polar, it's going to have to help it. And it helps that by having integral proteins. Integral proteins can fall under a couple of categories. They can be channels and they can be carriers. A carrier carries a very specific type of molecule. A channel is the one that looks like tunnels. They're based on size. If a molecule is small enough to fit through, it does. Cell membranes also have glycoproteins and glycolipids. These are for cell signaling and communication. It's not on the diagram here, but they have areas that are really rich in fatty acids. Those are lipid rafts. Again, it's thought, they're relatively new, but it's thought that those might be for cell signaling. The other thing that's here are peripheral proteins. Peripheral proteins are on either side of the membrane, and they're either structural or for enzymes. So again, this is everything we just went through. Integral proteins firmly attached. You can do that because an integral protein has both hydrophilic and hydrophobic portions to it, so that anchors it in the membrane. They can be channels, they can be carriers, they can even be receptors. Then we have peripheral proteins. They're loosely attached. They're more for supportive systems, and they're on either side of the membrane. Their function is to be enzymes or help move things in and out of the cell or link cells together. And then we have glycoproteins. They can help make something called a glycocalyx, and they're for cell-to-cell recognition. And again, lipid rafts, densely packed fatty acid areas, probably for cell signaling. All right, quick and fast. Other things cells have that we talked about in lecture. Cells have microvilli. Those are extensions of the plasma membrane. They are to increase surface area. You'll find them in tracts of the body that do a lot of absorption, like your small intestine. Cells are connected to each other by membrane junctions. If peripheral proteins connect them in such a way that they're kind of locked together and they do not share cytoplasm, that's called a tight junction. If the peripheral proteins lock them together, they still don't share cytoplasm. However, you can see this one looks really different. This one is made in such a way so that this connection is incredibly flexible and very hard to tear. That's a desmosome. The last of the three junction types occurs when proteins bind cells together and they allow the cells to share cytoplasm. That's a gap junction. Cells have organelles. Organelles can be membrane bound or non-membrane bound. I want you to pause the slide here, go through each of the organelles listed. You should be able to find it on the diagram, and you should be able to say what its function is. All right, cells have cytoskeleton as well. These are protein-based, and they're molecules that serve the function of either a tiny skeletal system or muscular system. Microtubules are just that, protein-based small tubes. They are very, very structural. We have motor proteins. Motor proteins physically move things across the cell. We have microfilaments, including myosin. Those are proteins that are contractile. Again, they help move things in the cell, and they help with phagocytosis and exocytosis and all of that. We have microtubule-based centrioles, always in the cell, but only used for cell division. And then combine microtubules and microfilaments in special ways, and you can get cilia or flagella. Cilia are the hair-like extensions, not microvilli. Hair-like extensions that beat in a wave-like motion to propel liquids and fluids and mucus. Flagella, making up words. Flagella have tails on them, and they allow a cell to swim. Now we look briefly at transport mechanisms. Again, we talk about them in lecture. If a cell moves a molecule with a gradient from high to low, and it does not use any kind of chemical energy to get that job done, that's called passive transport. And there's 4 kinds of passive transport. We have simple diffusion, facilitated diffusion, osmosis, and filtration. I'm going to define filtration separately. Diffusion is movement of solute molecules, movement of stuff, from high concentration areas to low concentration areas across the cell's membrane. And that's it. It doesn't even require that they go across the membrane. Diffusion itself just means from high concentration to low concentration. Cells diffuse inside them. In any case, molecules that can diffuse simply across the membrane have to be nonpolar because they have to be soluble across the membrane. Well, earlier we said things that are not soluble across the membrane, carbohydrates, proteins, DNA components, and RNA components. Those molecules cannot cross the membrane on their own, so you must help them with a protein. And if you help it with a protein, that's facilitated diffusion. So these are for molecules that are polar. Now we have to use channels and carriers, and especially with the case of a channel, we can be incredibly specific. The third example was osmosis. Osmosis is a specific case. It's the movement of water, or solvent, across a semi-permeable membrane. Water finds a way. Water can squeak across the membrane. Water also moves on its own special carrier proteins called aquaporins. Osmosis is related to the last two terms there, osmolarity and tonicity, and I'll define them later. So here's how it all works. Molecules that are lipid soluble are going to move from high concentration areas to low concentration areas, plain and simple. They can go across the membrane if they're lipid soluble because of the cholesterol that's stuck in there. Molecules that are not lipid soluble, so molecules that are polar, cannot cross the membrane on their own. Remember, they would hit the fatty acid tails on the inside and be repelled. So now we have to have integral proteins. This one is a carrier because it's carrying something very specific. This one is a channel. It's more like a tunnel, and it's just on size. If it's small enough to fit through, it crosses. This is water, so this is a diagram of our friend osmosis. Filtration is its own thing. Filtration is actually very different. Filtration is the diffusion of water and solutes through a membrane, but what drives it is a pressure gradient. You see the word pressure and gradient, that's filtration. So it's the movement of water and solutes from high pressure to low pressure. You do filtration in your kidneys, and you'll talk about it at length in Anatomy and Physiology 2 when you learn about the urinary system. All right, the other side of the story. Let's say a cell has to move a lot of something, or something that's really, really big, or it has to move something against a gradient from low to high. It wants to stockpile. In any of those cases, it's forced to use active transport.